Hi, welcome. Welcome to the 87th Cold Spring Harbor Symposium on Quantitative Biology. This year's topic, stem cells. And I'm very pleased to have Magdalena Gotz with me from University of Munich.
Yes.
And you just spoke this morning, Magdalena, and you were talking about organelles in different cell types. Is that essentially what your topic was?
Yes, so we think we have discovered a new principle of regulating cell function and by that also affecting development and disease. And this is that organelles that occur in all the cells, like the centrosomes or the mitochondria, they actually differ profoundly in their composition and henceforth then also their function between cells. So we had a science paper last year showing that the centrosome is actually changed by 50% between neural stem cells, human ones we explored, and neurons. And that was completely unexpected because people thought centrosomes do the same in all the cells and hence they are also similar in all the cells.
What you just said changed by 50% in what way? In volume, size, number?
No, proteome. So 50% of the proteins are exchanged. So it's really a profoundly different organelle, if you want, in one cell type versus the other.
And is that true then of mitochondria and the nucleolus and other?
Exactly. So the mitochondria we also profiled, we and others also profiled them, and also found massive differences. For example, between neurons and astrocytes, it's about a fifth of the mitochondrial proteome. So again, each fifth of the mitochondrial proteins is changing when you go from an astrocyte to a neuron, which is what we do, for example, in direct neuronal reprogramming.
And that begs the question, of course, why do these changes happen? Or what's
exactly, what's the function
of them?
Yeah, exactly. So for the centrosome, for example, we could show that many of the proteins that are specific for the neural stem cells are actually at parts of the centrosome where the cytoskeleton is anchored. So for the experts, the subdistal appendages or pericentriolar material. And indeed, they fine-tune the function of the cytoskeleton with profound disease-relevant phenotypes if this fine-tuning doesn't work. But just to say, that's one main function in the neural stem cells. But then in the neurons, most of these proteins are gone, and instead we have proteins at other hubs of the centrosome, at other places of the centrosome, the precise function of which we still have to explore, we in the field.
Well, I mean, I was brought up just to think that organelles were organelles.
Yeah.
A mitochondria in one cell did the same things as a mitochondria in any other cell, and they have the same composition.
Yeah, so that's
this is very much a dynamic view of what these things are doing.
And indeed, also, let's come back to the mitochondria. I mean, there one should say we already knew that in some cells they perform other functions, like in the astrocytes, they mediate lipid biogenesis. Neurons cannot do so. So the mitochondria as metabolic hubs, really, functional differences were already known. And indeed, we find that in the astrocyte proteome, we find, of course, these proteins that perform this function, and they are not there in the neuronal mitochondrial proteome. So let's say there the picture already was revised in terms of partial metabolic function, but again the extent of reshuffling of the total composition of proteins is quite mind-boggling.
And when you make iPS cells and differentiate them into neurons, neuronal or let's just say differentiate them into different cell types, the mitochondria, the centrosomes of these changes you've just described, they are parallel during this process of differentiation in vitro.
Yes, exactly. So actually we use an iPS-derived system, the human neural stem cells and the human neuron, to profile the centrosome. So indeed, these are data from human iPS-derived cells, because one needs lots of materials to do so, and that is a great system to do so. But yes, so in this in vitro differentiation, this exchange of half the centrosome proteome occurs from neural stem cells to neuronal differentiation.
And you've just mentioned a couple of times about the disease relevance of this finding.
Yes, because what we could then also show in this paper published last year was that we see a lot of RNA binding proteins at the centrosome, and amongst them also many common splicing factors. So splicing factors you would normally expect in the nucleus, right? But we find quite a lot of them at the centrosome. Amongst them, then we picked one disease candidate, namely a ubiquitous protein, which is expressed in all the cells of the body and also present at the protein in all the cells of the body, mostly in the nucleus, in the speckles for mediating splicing. But only amongst the examined cells so far, only in the neural cells, we found it at the centrosome. Now, indeed, when it's mutated in patients, the phenotype is a brain phenotype. Namely, the neurons get stuck in the inner part of the brain, let's say, close to the ventricle, which is why this disease is called periventricular heterotopia. And we could at least establish the first insights into a potential mechanism, why a common splicing factor mediates then this disease phenotype only in the brain and not in the kidney, because only in the brain it's at the centrosome. But what does it do there? It actually brings its splicing targets, we have shown it only for one, of course, to the centrosome, and this splicing target, when mis-spliced, when not spliced properly, indeed is causative for interfering with the migration of the neurons, and PRPF6 brings the RNA of this enzyme to the centrosome, and this enzyme actually phosphorylates microtubule-associated proteins. So we think that a splicing factor can act at the centrosome by bringing its targets to there, and they are ready to be locally translated and perform their specific function there.
So is the splicing done at the centrosome, or does the splicing already happen? It's simply the protein is transporting? Or
this we have not yet explored. All I can say is that we find a lot of the splicing machinery, also so-called core components, also at the centrosome. But we have no proof that splicing would occur at the centrosome, but it's amongst one of the functions that I think could be explored, but the more conservative interpretation would therefore be the one that I propose, that it simply brings its RNA with it after splicing. But this is still to be explored.
Are there any other examples of a splicing factor bringing its RNA?
Not yet, but
else?
Of course we are looking at more of these proteins, because in fact this is the top geoterms at the centrosome are splicing and RNA processing. So this is the highest proportion of proteins that you find there in the neural cells after we profile them. But yeah, we have looked only at this one. It's known that RNA is at the centrosome since ages. And it's also known from beautiful work of colleagues who screened, for example, for hundreds of RNAs encoding for centrosome proteins, and found that about 5% are present as RNA at the centrosome, but 95% not. So no idea at the moment what this specificity means. Why are some RNAs there, other RNAs not there? The proteins are anyway there. So there is a whole new world that we need to explore and understand, which is very exciting.
Well, I think we should stop at that point on a nice high note.
Okay.
Thank you very much.